Hi, thanks for coming. I'll be presenting on lineage program, which is a technique that we've developed to handle lineage-associated expression experiments, which is a particular type of expression measurement. So I'm going to start off with 3 questions that I'll eventually answer. And the first one is, can lineage information help model gene expression? So very broadly, the figure being shown on the slide is our current differentiation protocol. We start with the embryonic stem cell, and by combination of a variety of small molecules and transcription factors, we can push this along development into a pancreatic progenitor or intestinal endoderm. And we take expression measurements along this path. And the question is, if we have the information of who or of what cells they were derived from, can we better model expression? The second question is, can we identify developmental states and what we consider to be key cellular states during development? So given that we start with this structured experiment, can we identify which one of these states are really important? So in this toy example, on the left we have 3 titrations, and on the right we have the addition of a different compound. Can we identify the fact that the 3 different states on the left are actually the same cellular phenotype while identifying the separate phenotype? And the last question is, can we use a small number of co-expressed and co-regulated gene sets to explain overall gene expression throughout the lineage? So I'm going to be using the term gene program, which will refer to a set of co-expressed and co-regulated genes that occur throughout development. When we talk about embryonic development, we like to talk about stories of what's going on. We start with the embryonic stem cell, we talk about the downregulation of the embryonic stem cell factors and the upregulation of pre-endodermal factors. And then there's a suppression of alternative sulfates, activation of other endodermal markers. So we like to think of each one of these gene sets and these markers as a particular gene program. And can we decompose this lineage into a small number of these gene programs? Those are the questions that we're really interested in. And these questions can't really be answered with the current techniques that are sort of prevalent. The major techniques for finding gene programs are either linear algebra-based, principal component analysis, or non-negative matrix factorization. These allow us to decompose a set of expression into gene sets, combinations of gene sets. And latent Dirichlet allocation has a similar role in decomposing gene expression. But these completely discard lineage structure, and so they don't use the statistical information, and they also don't tell us what the key cellular states are. Clustering models can tell us what the key states are, but on the other hand, they don't give us a gene program decomposition. So I'm going to be describing our method through this toy example, where there are 2 cellular states, base state and state 1, and there are 2 genes, blue and red. Our model is going to parameterize the log fold change in expression between each of these states. The generative model for these is going to be the base state expression plus sampling noise, which we model to be multinomial. The latent states, which we don't infer, will be parameterized by log fold changes. In order to use the fact that we have some structure, we're going to be using sparse regularization techniques in order to induce structure in our overall solution. We're going to be using 3 different kinds of regularization. First, we're going to try to force the total number of expression changes throughout the lineage to be small. Next, we're going to try to have few unique cell states. And lastly, we're going to try to have few gene programs. And this is all both for the sake of interpretability and to avoid overfitting. And we're going to use L1-based regularizers throughout. So in order to decrease the number of total expression changes, we use lasso penalties over individual gene expression change events. So in this toy example, we have the blue gene changing small or slightly being downregulated, and the red gene being greatly upregulated. Addition of small amounts of lasso penalty will force the blue gene to snap to its previous expression level, causing its fold change to be 0. This type of regularization allows us to control individual gene expression changes. The second regularization penalty that we'll add will be the fused lasso, which penalizes the total norm difference between 2 states. This is distinct because it does not penalize individual genes, but rather changes between states. So in this new toy example, both of the genes are slightly downregulated, but the fused lasso forces these 2 states to be identical. So this can be considered a form of tree-based clustering. And lastly, and the most complex, we're going to control the total number of programs and therefore the total number of overall degrees of freedom using the trace norm penalty. What we're doing is we're creating an implicit factor decomposition. Every time, or every update of the model, we're going to be keeping this matrix decomposition, UDV, where U represents the set of gene sets, D represents the strength of each of these gene programs, and V represents where these gene programs are used throughout the lineage. So we have this following decomposition that gives us gene sets, program strengths, and program usages. By using the lasso penalty on the program strengths, we can recover a small number of these programs that are non-zero. And the reason why we're going through this sparse penalization framework is because this makes the model inference extremely elegant. The regularized objective is overall convex non-differentiable. What this means is that we can guarantee that the solution we get is unique. We don't have to do random restarts. It's extremely fast to fit this. You can look at the paper for the particular proximal operators we use, but the model fit is very fast, and we can actually enumerate all of the possible regularization parameters. And so I'll show results on a very large pancreatic and combined neural lineage that we've collected over a large time span. We have a set of 105 microarrays. 88 of these are pancreatic. Those are the red experiments shown there. And 17 of these are motor neuron, shown in blue. We have 24,000 genes being measured. And we're going to try to reduce this nearly 100-state lineage into a very small number of key cellular states and key developmental programs. We're going to be using 3 evaluations to predict our model. The first one is going to be how well we predict held out data. This is a very basic model fit question. The second one is going to be how well do our programs match known developmental marker sets. And lastly, we're going to compare our, the cell states that we eventually learn with hand annotations of this same lineage. So first, throughout we're using cross-validation to select the optimal number of parameters. And what may be surprising is that the optimal number of edges and programs are both extremely sparse. What's shown on this slide is the number of edges and the number of programs are log likelihood as a function of number of edges on the left and number of programs on the right. And in both cases, the optimal solution, shown in blue, is extremely sparse. And all of the sparse solutions have relatively high log likelihood. What this shows is that despite the very high complexity of the tree being shown before, a very few gene sets and a very small number of edges are sufficient to explain the entirety of these changes. And compared with fully parameterized models such as SVD, LDA, and non-negative matrix factorization, on held out data, we're doing 140% better in terms of squared error. This shows that our model is avoiding overfitting that is very prevalent in SVD and LDA type methods. And something that is even more surprising, considering that our algorithm is sparse, is that despite having fewer non-zero genes, our algorithm manages to have higher geo-enrichment given a particular p-value. One way to interpret this is to think of our algorithm as a particular frequentist inference method. We can think of having a higher fraction of geoterm enriched per p-value as having higher power in terms of testing. This is because we're using the lineage information in order to model only the differences. And finally, what is sort of a more qualitative examination of our algorithm, we took the cellular states and we had our collaborator do an independent hand annotation of what were the key developmental states throughout our lineage. On the left here is the optimal learned cell tree, and on the right is our collaborator hand annotated cell tree. And we find that we separate the motor neuron and the pancreatic lineages, which is a very basic requirement for any algorithm. And for the motor neuron lineage, except for merging the day 2 and day 3 motor neuron states, we get perfect recovery. And on the right side, we get perfect recovery for the early stages of the pancreatic lineage. Day 3 mesoendoderm, day 4 preendoderm. These are both exactly matching. This is very good considering the fact that we started with over 100 states, we collapsed into a dozen, and we nearly exactly match hand annotated cell states. And finally, if we look at the particular gene programs that we pull out, we're pulling out fairly subtle marker genes that are known in development. So the numbers shown alongside the cellular states are the locations at which our gene programs are being used. The table below shows the particular gene programs. Going through these somewhat briefly, the first program, so the other thing is that we discovered two different types of programs. One which is shared across lineages, which we call global, and one which is constrained to a single lineage. So the two global programs that we find are first the loss of pluripotency, which we expect to be shared in both motor neuron and pancreatic lineage, as well as the suppression of alternate sulfates. Cardiac muscle, skeletal muscle, those types of sulfates are being suppressed in both paths, and this is being extracted. In terms of cell-specific programs, what's very impressive is that our algorithm managed to pull out the Hox positioning code as separate from the general motor neuron positioning. The Hox positioning code is shown in 3, and the general motor neuron specification is program 6 and 8. And so our algorithm is able to distinguish the fact that the Hox positioning is a temporary expression increase at day 4 motor neuron rather than the continual increase in expression for the other motor neuron markers. So our algorithm is able to pick up very subtle gene set markers that were not easily pulled out by simple differential gene set analysis. So returning to our original questions, can lineage information help model gene expression? I think the answer is yes. Looking at our held out error experiments, it's clear that in terms of squared error or in terms of model fit, lineage program significantly outperforms existing decomposition methods for modeling gene expression. And for the second question, can we identify key cell states during differentiation? I think that the 100 state to a 12 state collapse with nearly exact match to hand annotation shows that we're able to really recapitulate the key cellular states during a developmental experiment. And lastly, the question, can a small number of gene sets explain gene expression? This is more speculative, but looking at the gene sets, we've recovered what we would expect as the significant gene programs to be active during these lineages. Loss of pluripotency, specification of pancreatic and motor neuron lineages, positioning codes. Those types of things are being extracted and specified into the appropriate parts of the lineage. We've also made our code available on our website for use, and there's documentation. Our algorithm isn't just for lineage-specified models. It's easily extendable to other types of grid-based or other types of structured cellular models in which you know the derivation of your cells. Yeah.
I have a question about this definition of the key cell states. Have you looked at a large number of data sets to try and identify key programs that define these key cell states? So could one try and extract something like that?
So in terms of trying to map marker genes to particular cell states, so I guess the way in which these were defined is our collaborator looked at the key marker genes, defined the cell states. And in terms of our learn programs, they're similar. It is the case that because of the way our decomposition works, in order for there to be a distinct cellular state, there needs to be a very distinct program usage pattern. And so what that means is every distinct cell state is associated with at least a different program usage pattern, if not a distinct program. And so it is the case that a lot of these are associated with single gene sets.
Thank you for the talk. How does your algorithm perform with respect to isoforms and alternative splicing?
We assume that the expression values are given as sort of a black box. We don't really treat alternate splicing or even overlapping gene names and things like that.
Thank you.
So, in the example you show, you have 88 versus 13, the sample size.
Yes.
Do you have a minimum number of sample size in order for this to work?
So there is no minimum number of sample sizes for this algorithm to run. There's a very basic linear algebra constraint that the number of programs you extract will always be less than the number of conditions that you have. And so that's the basic constraint that we have. So if you only have 3 states, you can at most extract 2 programs. So our example is ideally suited for this type of thing where you have a lot of different states.
What is the role of heterogeneity of the initial data for your predictions? For example, here you use 3 types of cells, neurons, pancreatic, and embryonic stem cells. Instead of that, if you had used only neuronal cells, would it perform better or worse?
Right. So we have, in the actual proceedings paper, we have held out validations and things like that for just the pancreatic branch as opposed to the neural one. The reason why we have the neural one is because it makes the problem significantly harder for our model to deal with because there's such diverse or distinct differentiation lineages. But our algorithm does perform very similarly on both lineages. They extract similar cell states. They have similar held out error scores. So in terms of what happens when you add or remove branches, they degrade in a graceful way.
And if you just give it one cell type, like if you just give it embryonic stem cells only,
yes,
will it produce just one state, or will it try to create multiple states?
It can't merge, or it will only merge states. So if you start with one state, it will remain at one state, if that's your question.
Hi, great talk. I was wondering, I always hate this kind of question, but I remember a couple other algorithms, one from ISMB a couple years ago called mixed D-tree, and then there's another one from the Plevrides group called SPD, and they try to infer these hierarchies from expression data. So maybe the algorithms have different data requirements, but I was wondering if you guys thought about comparing to these other methods.
Yeah, if I remember what the mixed B-trees paper is about, it is, I think it clusters the cell states, or I think that's what it does, given or not given the lineage, which is a very distinct problem, I think.
So yours has to have the lineage.
Yes. So ours. So I think that paper was, given that there is some latent lineage we don't know, can we use that to do better? Our question is, given that we know the lineage, can we do better? Of course, we can remove the lineage and then feed it to their algorithm, and presumably it will do better than SVD. But I think the questions that it answers is it's very divergent.
Got it. Okay, thanks.
So kind of a follow-up to that. I'm not clear exactly how you specify the lineages. Like, would you, for instance, for the input to what you would pass to your model for this specific example, would you tell it, there are 2 branches, or what exactly do you tell it?
Right. So I guess this is our actual experimental tree. And so we have a set of experiments. So you feed it a graph. You also feed it your set of experiments, and you feed it a map telling where each of the expression measurements were taken. And then given that, it will collapse them appropriately.